Let's talk about DNA extraction or DNA purification. The general steps are the same with protein purification. The only difference is we must consider that your DNA is inside the nucleus. Furthermore, if you are considering eukaryotic DNA, then you also must think of a way to remove the histones on which your DNA is wrapped around if we want to obtain pure DNA samples. So again, the flow is the same. You lyse the cell wall or the cell membrane. You also lyse the nuclear membrane, and your nucleic acid comes out from the nucleus. So the outline is you have lysis. As you have learned from protein purification, we have several types of lysis. We have mechanical lysis, chemical lysis, enzymatic lysis. So the methods that you learned also apply for DNA extraction because we are just destroying the cell wall or the cell membrane. Sometimes, or most of the time, these lysis methods would also destroy the nuclear membrane or the nuclear envelope. Afterwards, we remove contaminants such as cell debris and proteins. So we add some reagents, such as your proteinase K, which is an enzyme that degrades proteins. After this, we allow DNA to precipitate through some chemical reagent. The goal of precipitation is not only to isolate the DNA molecules, but also to concentrate the DNA sample. So especially if we are to use the sample for a method that would require a high concentration of DNA. So the most common reagent used to precipitate DNA are alcohols. And then finally, we dissolve DNA in an appropriate buffer and use it for other methods or techniques that we want to perform. All of these steps are, of course, performed in the presence of an appropriate buffer. Your DNA extraction methods are classified into two general types. We have chemical DNA extraction methods and physical DNA extraction methods. Under physical DNA extraction methods, we currently have two subtypes: magnetic bead DNA extraction and paper DNA extraction. Under your chemical DNA extraction methods, we have organic and inorganic methods. And the oldest and classical method used to extract DNA is your phenol-chloroform DNA extraction method. Under the inorganic DNA extraction methods, we have the proteinase K DNA extraction method, salting out method, and silica gel-based DNA extraction method. I will discuss a few of these methods. First, let's go to the phenol-chloroform extraction method. The major components used in this extraction method are the buffer, phenol, chloroform, and isoamyl alcohol. Of course, the function of the buffer is to maintain the pH of the solution so that your DNA will not denature. In your buffer solution, you have tris, EDTA or EDTA, SDS, sodium chloride, and magnesium chloride. As you all know, tris is the reagent that gives the solution a buffering capacity. EDTA or EDTA is a chelating agent, and it prevents the action of enzymes that degrade DNA. So whenever you perform DNA extraction or DNA isolation, it is important to have EDTA in your solution. SDS or sodium dodecyl sulfate, you have encountered this in SDS page and learned that this is a detergent. So SDS helps in the lysis of your cell membrane, the destruction of your nuclear membrane, and also in the degradation of some proteins. Sodium chloride provides the right tonicity of the solution. Aside from that, it also gives the correct ionic strength of the solution to maintain the structure of your DNA. Sodium chloride will also play an important part in the precipitation step. Magnesium chloride provides magnesium ions that helps in stabilizing the structure of your DNA by binding to the phosphates in the phosphate sugar backbone. Remember, the phosphates are negatively charged in physiological pH, so the magnesium ions helps to stabilize the negative charge of your phosphate sugar backbone. The phenol used in phenol-chloroform extraction performs two major functions. Number one, phenol helps in the denaturation of proteins, thereby preventing them from contaminating our DNA sample. Second, and the more important function played by phenol is in the extraction of the DNA molecules. Phenol is immiscible with water. So by using phenol in this DNA extraction method, we are actually taking advantage of another technique called liquid-liquid extraction. When we add phenol to an aqueous solution containing DNA, the result is a solution with two layers, the phenol layer in the bottom and the aqueous layer in the top. This is because phenol is more dense than water. This step enhances the purity of our DNA sample. DNA remains soluble in water, so it will stay in the aqueous layer. Meanwhile, denatured proteins will tend to go to the organic layer because their hydrophobic regions are exposed when they are denatured. Chloroform enhances this effect of phenol. There is no specific explanation as to how chloroform enhances the effect of phenol, but it has been found to be effective. So after removing the phenol layer, we add the alcohol for the precipitation step. Later on, we will learn the principle behind the precipitation of DNA in the presence of alcohol. The next type of DNA extraction is enzymatic extraction. The important reagents, of course, we have our buffer, and we have proteinase K, and again, we have isoamyl alcohol. The components of our buffer are tris, EDTA, SDS, and sodium chloride. Notice that we don't have magnesium chloride in our buffer. Magnesium chloride can actually interfere with the action of proteinase K. Proteinase K is an enzyme that can degrade proteins. So enzymatic extraction actually increases the purity of a DNA sample. Its disadvantage, however, it's a longer technique. So it takes on average 2 hours to incubate your DNA sample with proteinase K before you proceed to the precipitation step. Next, we have the silica-column based DNA extraction. If you are to join a well-funded laboratory, you would find that they have DNA extraction kits, which are very convenient because it allows you to isolate DNA, let's say from E. coli, within 30 to 45 minutes. The principle here is that you have a small column made up of silica gel, DNA in the presence of chaotropic salts, such as guanidinium chloride, will have affinity to silica. The effect of guanidinium chloride is not yet fully understood, but they found out that this salt increases the affinity of DNA to silica. Furthermore, in this method, sodium chloride is also important. The cation, sodium from sodium chloride, also enhances the affinity of the DNA molecules to silica. Take note, silica is negatively charged. Your DNA molecules are also negatively charged due to the phosphate groups. One most likely explanation for the action of sodium from sodium chloride is that the sodium ions first interact with the silica. And then the DNA molecules will now have affinity to the sodium ions attached to the silica. So the sodium ions act like a bridge between the silica in the column and the DNA molecules, allowing purification of DNA. So the general flow is that you allow your sample, let's say your lysate, to flow through the column, spend some time for it to adhere to the silica gel, wash the column. This is to remove other debris that might have very weak interactions with the silica. And then finally, elute the DNA from the silica column. Next, we have magnetic bead extraction, which is more expensive, probably the most expensive of all DNA extraction methods, but is the fastest and most convenient. So the principle behind magnetic bead extraction is that you have beads or magnetic beads whose surface are chemically modified. Anything that you want to purify must have affinity to these surface modifications. For example, mRNA molecules are synthesized with what is called poly-A tails. This means that at the 3' end of mRNA molecules, you have a long sequence of adenosine residues. Since we know that adenosine interacts with thymidine, then we can modify the surface of our magnetic beads by attaching polythymidine strands. So if we introduce our sample, if we introduce these magnetic beads to our sample, then the mRNA molecules will now attach themselves to this polythymidine strands. And then we expose the solution to a magnet. Since the beads are attracted to the magnet, they will stick to one side of the tube, and we can easily pipette out the solution, removing all unwanted molecules. And then dissolve the isolated DNA, or in this case, mRNA molecules, in the appropriate buffer, elute it from the magnetic beads, and now you have a very pure sample of mRNA molecules. Finally, we go to alcohol precipitation of DNA. When your DNA is dissolved in water, most of the water molecules also interact with the phosphates in your DNA molecule. You know that water molecules are polar, so their positive end will interact with the negative charge of the phosphate groups. Remember too that you have sodium ions in solution coming from sodium chloride. Your sodium ions will also interact with the negative charges of the phosphate groups. However, because of the high dielectric constant of water as a solvent, the sodium or the positive charge of the sodium ions and the negative charge of the phosphate groups in the DNA do not feel each other's charges, meaning they have very weak interaction to the point that it's possible that they don't feel the presence of each other in water. But if we add alcohol such as ethanol or isoamyl alcohol, this solvent has a lower dielectric constant than water. This means in the presence of alcohol, the sodium ions can now feel the negative charges of the phosphate groups. Since they can feel the negative charges more, they can now form ionic interactions. The effect is that the negative charge of the phosphate groups are neutralized. And you know that neutral molecules tend to precipitate out of solution. So this is the principle behind alcohol precipitation of DNA. You can explore a virtual simulation of DNA extraction from this website. It also contains discussion explaining the principles behind each step and will also allow you to visualize how the method is carried out.